Merle Patterning and the Genetics Behind It by Dr. Merlin Merli. Well, in 2006, Clark and colleagues performed a whole genome scan on Shetland sheepdogs and made a startling discovery. They found a linkage disequilibrium via microsatellite marker on canine chromosome 10 in Merle dogs. The region where the marker was located is known as the SILV gene. The SILV gene is known to be important in mammalian pigmentation, playing an important role in melanin production and deposition. It has even been suggested to be involved in the biogenesis of premelanosomes. This evidence suggested that the SILV gene was indeed responsible for the Merle color. Isn't that wonderful? Anyways, they found a short interspersed element, also known as a sign, inserted at the intron 10 exon 11 boundary. A sign is a retrotransposon, and researchers found that it's segregated with the Merle phenotype in multiple breeds, but was absent in dogs representing breeds that do not have the Merle pattern. The presence of the sign in the gene results in disruption to gene expression and leads to the diluted hair pattern and other impairments that we see. To help explain the impact on gene expression, let's look at this image. In A, the sign is located within an intron, resulting in no damage. B has the sign located near a regulatory region and results in either an increase or decrease in gene expression. If you look at C, the sign is located within an exon, which disrupts the open reading frame and results in a non-functional protein. Lastly, D illustrates the situation where the sign inserts at an intron-exon boundary. This alters the protein product and creates a situation similar to C. In our case, we are dealing with situation D. It's important to notice the PCR-identified products of the SILV gene for Merle and non-Merle dogs. The size of the product differs greatly due to the sign insertion. Therefore, the sign insertion plays an important role in the phenotypic diversity of dog breeds. You may be wondering where these signs came from. Interestingly, the mobility of signs in the canine lineage is even higher than that in the human lineage. But why is sign retrotransposition so active in the dog genome? Well, it is thought that repeated demographic crashes in the dog population occurred near the time of divergence between wolf and dog, resulting in genetic drift and reduced efficiency of selection against deleterious signs. We have seen evidence to support this, as the sign insertion in the SILV gene is responsible for the Merle phenotype in many breeds of dogs, an indication that the sign insertion occurred before the divergence of breeds. You guys still following all this? Well, try to follow the rest. As I am sure you have found out already, deafness is commonly seen in dogs with one or two Merle alleles. The type of deafness is hereditary congenital sensory neural. Degeneration of the vascular supply, called the stria vascularis, to the cochlear duct of the inner ear is seen in this condition. Secondary degeneration of the cochlear hair cells and neurons will occur. It is thought that the degeneration occurs due to the absence of melanocytes, since previous data has shown that melanocytes are essential for normal hearing function. Histological studies in mouse mutants and deaf Dalmatians demonstrated the absence of strial melanocyte-like cells, which appear to be vital for normal strial development. Research also noted that the melanocytes were responsible for maintaining the sodium-potassium potentials. A disturbance to the electrical potential interferes with the individual's ability to conduct sound. The picture on the left shows the position of the inner ear, while the image on the right illustrates the location of the blood supply in the cochlea. Other than hearing impairments, dogs that are considered double Merles have other abnormalities. The structure of the eye can be compromised, resulting in functional impairments. The types of disorders can be seen here. Other developmental defects have been reported, sometimes to the degree of being incompatible with life. Some other interesting pieces of information that are worth mentioning include the harlequin color found in Great Danes. Merle is not a breed standard acceptable color in Great Danes, but harlequin is. However, the harlequin locus interacts with the Merle locus, and harlequin dogs carry the same insertion that we have just seen in other breeds. Also, Waardenburg syndrome is a result of mutations in several genes involved in the development, migration, and differentiation of melanocytes. One of these is the MITF gene, which happens to be a regulator of SILV. Now you can understand more fully the link between Waardenburg syndrome and the Merle condition, and see how important it is to study this specific retrotransposon, as it provides an excellent model for which to study similar human disorders. I hope that this information was helpful and you have a safe trip home.
Justin! Justin! Get over here! Sit! Can't you be like Turtle?
Hi, Amanda.
Wow, took you long enough.
Well, that's kind of a long story. But we did find out that the problems you're having with Justin are probably because he's a double Merle.
A what?
Well, so why don't we go sit down and discuss our options?
I'd appreciate that. So how does this affect Turtle? I was really hoping to breed her and get some Merle puppies.
Okay, well if you're looking to breed Turtle, your best bet would be to breed her to a solid coated dog and not another Merle.